Chapter 17, the conclusion for this term, looks at biotechnology and genomics, and it really is a summary of a lot of the information that we have already taken a look at, as well as a look into some of the very new and exciting applications. So the very first one that we're going to take a look at is the ability to remove and isolate DNA and RNA. So the way this is done is that first we use a detergent to break down the lipids or the fats that might be in a collection of cells or tissues. After the cells are broken apart, and so the contents of them have been released, we can use some enzymes like proteases, which break down proteins, and ribonucleases, which break down RNA, to remove those materials from the sample. Next, we will have to centrifuge the material and remove either the DNA or the RNA that we're looking for, which will be contained in the liquid or the supernatant above. Once we have isolated that, then we can add some ethanol in, and that will create strands of DNA or RNA, depending on which we're collecting, and we can isolate those. This may be a technique that you have actually done at some point in a high school lab. So here is a picture from the text which is showing that procedure. So any debris, cell material, broken down proteins, broken down RNA is in the material here, and then the supernatant, which is the liquid portion that contains your DNA, we add ethanol to that, and that will cause the DNA to make strands, and you can actually isolate it. The next technique we're going to take a look at is that important technique we've already looked at in lab called gel electrophoresis, where we can take DNA fragments and compare them to a known DNA profile. So if we have these fragments that are of various sizes, we can use a gel material in order to separate them by placing them in a charged field that has a negative end and a positive end. So we add the DNA to the negative end, and it's going to move towards the positive end through the agarose gel. And that gel has pores in it, which will sort the fragments by size, causing them to go through. The smaller fragments go through the gel more easily than the larger ones, and the end result is that you can see a distribution of the fragments based on size, and you can actually collect them. You can cut out the DNA from the gel and isolate just the segments that you're interested in. And here is a picture again of performing that DNA fragment separation, and you can see you can compare them to a known DNA ladder, which has fragments of various sizes. The next important technique we're going to take a look at is called polymerase chain reaction. And in polymerase chain reaction, we are going to target in on a particular DNA region that we want to make more copies of. So PCR is used to make more copies of a specific region of the DNA. So you might have, say, only a small amount of the DNA and you want to make more of it. First, we need to use regions or short pieces of DNA that match up with the segment you're trying to reproduce, and these are called primers. So we need to have those, and also we're going to combine these primers, which will tag on to the DNA that you're trying to copy. We're then going to add in another enzyme called Taq polymerase, and we will add in the units, the deoxynucleotides, because we want to be building more DNA units, more fragments. So that Taq polymerase, so this is a bacterial enzyme that was isolated from a very interesting species of bacteria that are able, they're found in hot geysers, so they're able to withstand very high temperatures, and so the temperatures that are needed in the polymerase chain reaction are quite warm, and so we can use that this particular enzyme to help us build more DNA fragments. So here is a diagram representing the PCR, and as long as you get just a sort of basic understanding that the first thing that you need to do is denature your sample. So separate out the two strands. You notice we have the 5' to 3' and the 3' to 5' strands. We're going to separate those out. Then the step 2 is called annealing. So annealing is when you add the primer and it sticks on to your sample. Remember, the DNA is going to be added 5' to 3', so we're going to go in different directions. Step 3 is our DNA synthesis, and here's where we're going to add in our Taq polymerase, which is going to make our new strands as it recognizes the primer, which are these, again, these green bars. We're going to repeat these steps in various cycles. We're going to do more and more cycles, which are going to make more and more strands, and then we'll be able to isolate these strands and have a whole lot of DNA, which is of the particular sequence that we're looking for. Our next technique is called blotting, and there's a number of different ones of these. There's southern blotting, northern blotting, and western blotting. We'll talk about the differences here, but we'll start first with the southern blotting. And the purpose of southern blotting is, again, to isolate and recognize a sequence of DNA. We start off using a gel electrophoresis, and here when we have an entire sample of DNA but we're just looking for one particular piece, then we run the whole sample, but we might see that there's so many fragments that it's really hard to find the one that we're looking for. So we can then take and add the regions that are of particular interest to us to a new nylon membrane. And this nylon membrane will allow us to add then to it some probes, which are short little pieces of DNA that mark just the sequence that we're interested in. And these probes will be labeled with a fluorescent dye, and so that way we'll be able to visualize them under a UV light. So the DNA fragments are separated on the gel, they get transferred to the nylon membrane, and then we add in the probes so we see just the DNA that we are particularly interested in. So that's a southern blot, and this is the blotting when you add it to the nylon membrane. And a northern blot has the same procedure, but in this case we're isolating RNA from the gel. And a western blot, you're doing a run of proteins on the gel, and then you're using antibodies which will bind to those particular proteins that will tell you which are the particular sequences of interest. Our next topic is moving on to cloning, and we're going to talk about molecular cloning and then reproductive cloning. So anytime we use the word cloning, we are copying DNA. But in molecular cloning, we're only copying a short region of DNA, and then we're going to utilize that region in order to add that in to another species. So here we have what we're going to utilize are bacterial plasmids And these are little small pieces of DNA that are found in bacteria, and they can copy themselves without the chromosomal DNA. So they have regions in them called multiple cloning sites, or MCS, and these are regions that can be recognized by a specific enzyme called a restriction endonuclease. And the endonuclease is going to cut open the plasmid at these positions. So a restriction endonuclease will recognize those sites that I just mentioned and add them up. So it's an enzyme which is going to cut open the plasmid at a particular spot, and then it leaves a little bit of an overhang. We'll see this in just a minute in the diagram. And these are ends which will allow a new piece of DNA to be added in. So these ends are called sticky ends. And so we'll be able to splice in a new piece of DNA. So here's our diagram, which is showing the process. We have our plasmids, which are in these blue circles. And here we have a piece of DNA that we want to add in to the plasmid. So here we have the site that we're going to break open. That's our MCS. We're going to break open this site using the restriction endonuclease. So the enzyme has opened up this site in the plasmid, and then we're going to add in the region of interest. We will isolate that from our DNA, and because they have the sticky ends, then it's going to fit right in. Then this bacteria, we're going to add these plasmids to a sample of bacteria which may or may not take up the plasmid with the special new insert. And we'll always have to go back and check that particular situation if it was added in. So this new region is now called recombinant DNA because we have added new DNA into a hopefully a new another organism, a bacterial organism, and any proteins that are made from this new DNA, which doesn't belong to the bacteria, would be called recombinant proteins. Here's a short video which will again go over that process. Now, where molecular cloning was just adding a short region of DNA, here when we talk about reproductive cloning, we're talking about taking a complete genome from an organism and creating an entire new multicellular organism. So the way this is done is that you can take an egg cell, and remember, an egg, an oocyte, will have a haploid nucleus that's waiting for the sperm. But if you remove that haploid nucleus, so you remove the DNA that was part of the egg, and all you have is the egg's machinery left, and you add in a fully diploid nucleus from some cell of an organism that you want to make another copy of, then that egg will think that a sperm has bound to its nucleus and created a diploid nucleus, but in fact you've replaced it. It will go on to form a zygote, which is the first cell of a new organism, and it can go on to then divide and create a whole new organism, which is a genetic clone of the one from which the nucleus was obtained. So this was first done in sheep back in the 1980s, and we created Dolly was the very first cloned organism, and so a cloned mammal. So here we have a sheep, and we have taken an egg from this sheep, removed its nucleus, so enucleation. Then we took some cells from a different breed of sheep, and we just took cells, not germ cells, regular cells, And from this, we would take the nucleus from one of these cells. We would add this nucleus in with this egg and create a zygote, which could go on to form a growing embryo. We could then put it into a surrogate. So this organism did not donate any genetic material, and let it grow, and we will see that the resulting organism, Dolly, this sheep, does not look like its mother that gave birth to it because it has no genetic material from this organism. Our next technique, which is really fascinating, is called genetic engineering. So genetic engineering is the process where we take and make use of that recombinant DNA technology we talked about before. So we can actually use new pieces of DNA and introduce them into an organism and create new techniques and new organisms that can be beneficial to living things. So we do this using recombinant DNA vectors, either bacteria or viruses. And we're going to add in the foreign DNA using these recombinant DNA vectors. So remember, a recombinant DNA vector means we've taken a bacteria, a plasmid, or a virus, and we have added in a piece of DNA that's of interest to us that we want to make more copies of. So say we have a specific piece of DNA which we want to add into another organism to increase or improve that organism. So one example is that there are certain proteins that are present in fish that live in Arctic areas that keep them from freezing. So these are proteins that are made because of the DNA in them. And there are problems with strawberry plants that if a strawberry plant freezes, then that strawberry plant is no longer functional. So wouldn't it be great if we could take the ability to make these proteins that the fish use and inject them into strawberry plants? So then therefore strawberry plants would be more resistant to problems with freezing. That would be an example of using genetic engineering in order to make something better. So this process is what we call producing a genetically modified organism, or a GMO. So the organism is also called transgenic because it possesses DNA from another species. And there are lots of applications that we can use this recombinant DNA technology for. We can use it to make lots of antibiotics. So we can harness the use of fungi to make antibiotics. We can insert a particular piece of DNA, which will cause them to make more antibiotics for our use. Vaccines, this is how we make mass production of vaccines. We can harness the use of bacteria to create the various vaccines that we need. Hormones, this was used, particularly a good example is the production of insulin. So up until 1978, anyone who was diabetic who needed insulin, we had to isolate it from pigs, the actual insulin hormone. And this would cause problems because this was pig insulin, not human insulin, or could cause problems. But then in 1978, we used recombinant DNA to insert human genes to make insulin into bacteria through plasmids, and now we produce recombinant insulin, which we can make lots and lots of, and it's actually human insulin that's being made by these bacteria. So there's no longer the issue of having a reaction to the insulin. And also we can make different crop plants which are, say, resistant to temperature, like I mentioned before, or resistant to certain pesticides. And so these crop plants are what we call GMOs. Another great application of genetic engineering is being able to cure a disease using gene therapy. So here we can use a vector. In this diagram we're showing a viral vector. We'll transform this virus so it doesn't actually cause any disease. We'll use a virus that doesn't cause a disease itself, but we will inject into this a new copy of a corrected gene, and we will apply this to a patient who has a disorder, a genetic disorder. We will introduce a new gene into this patient to hopefully correct a genetic problem. The next section of the chapter talks about mapping genomes, and it's important to remember that we have introduced some of this information before. So genomics is when we study an entire genome, so all the genes, all their nucleotide sequences, and the interactions. So this is everything about a set of DNA in a particular species. We can then generate a genetic map. So we've talked about genetic maps. This is the location of the genes on every chromosome. So we can find out where all the genes are and notice their differences and how far apart they are from one another. We can also make use of genetic markers, things like single nucleotide polymorphisms. So these are repeated sequences within the genome that we can use as an identification marker. There's other markers as well, but the single nucleotide polymorphisms are the only ones that we're going to introduce right now. So we've seen this picture of chromosomes crossing over, and this indicates where genes are linked, remember, and where they're not linked. So any genes that remain on a single chromosome are linked. Where they cross over, we're now creating new recombinant chromosomes that have new linked genes. So here's a genetic map showing where all the chromosomes are located. We also can look at a physical map to see how far apart the genes are and how many genes and what their nucleotides are. And we can do this through a physical mapping. So all of this mapping leads to the fact that once we know what the genes are, where they're located, what the nucleotides are, we can then apply these genomics, these mappings, to a whole host of different possibilities. Things like personalized medicine, where if we knew exactly what your genes are, we could provide for you exactly targeted therapies and medicines. And there's, again, a whole list of them. We can predict disease risk, look at drug interactions. We could study more microorganisms like bacteria. If we know all their genes, we can create biofuels. We can look at assessment using mitochondrial DNA, which is the basis for all these 23andMe type tests. We could use it to solve crimes, which is forensics, and we can use it to improve agriculture or our production of food crops. So one of these applications is called pharmacogenomics. Pharmacogenomics is looking at a gene sequence using genetic mapping for an individual, and then we can apply particular therapies to that individual. So this may eliminate any problems that that individual might have with the fact that they may not respond to a particular treatment. So we would know ahead of time that they wouldn't respond to a particular drug, and we could maybe use a different drug. So this would mean that they would have a physical response to a particular drug, and we would want to give them only the one that's going to cause their genes to express and produce the desired results. This will also remove any potential toxic side effects that might happen from a drug based on their particular gene sequences. So genomics are looking at the gene sequences in an individual, but proteomics are the study of the proteome, which is the set of proteins that a particular individual might make under certain environmental conditions. So this relies on us being able to analyze proteins that are produced in certain situations. It's a way to study different types of cancer. So let's take a look at cancer proteomics. So we can look at a particular cancer patient. We can look at the proteins that they can make during their cancer disease, and we can help to get a good, proper treatment plan for them. So we can look at biomarkers. And so this is already done for PSA or prostate cancer treatment. So we look at proteins that are made in prostate cancer, and this can help indicate whether an individual, if they have elevated PSA, is possibly to be diagnosed with prostate cancer. We can also look at protein signatures, and we can find out which particular version of a cancer might be being present in an individual by looking at which proteins are being produced in that particular type of cancer. And so this would allow us to better treat individuals based on what their signature results are. So biotechnology is an incredibly exciting and new application of the modern side of biology.